we're going to be talking about the structure of DNA. How is it that the DNA can actually fit into such a small, minute volume of nucleus as to it would require some hierarchical organization, and I will actually walk you through as to how this hierarchical organization is set. So, having said that, I would like to start a little bit basic so that I can complete the whole picture, because I do not know how much of this kind of information the students have in Tehran. I know that people here, in my students, I do cover these back and forth. So I will go a little bit basic, so don't worry, it will get a little bit more difficult as I go along. So we talk about bonding structures regarding the chemistry of DNA. So here we need to pay attention to two major bonding. Number one is the covalent bonding, okay? And number two is the hydrogen bonding. As compared to hydrogen bonding, covalent bonding is an electron sharing between atoms, and these can be formed and broken through chemical reactions. They are actually stronger bonds, obviously, compared to the weaker bonds of hydrogen bonding. And we have examples like these just to give you an idea what I'm talking about. On the left-hand side, you see a double and triple bonds of covalent bonding, and throughout the study of DNA itself, and time to time, I will come and then touch base upon this. So I felt the need to actually introduce these kind of information to you to be able to have a strong foundation as we build on information here. So on the right-hand side, you see hydrogen bonding. You can see that it's clearly the hydrogen bonding is being made between oxygen molecules here, weaker bonds. That's why the reason you can get three different stages of water. You can get like liquid, solid, and gas. Now, although we are actually taking into consideration regarding the information about DNA as the molecular biology and genetics concept, it's a multidisciplinary thing to talk about DNA, because it not only involves molecular biology and genetics, but chemistry, math, and physics as well. So I will touch base on the basic chemistry of the nucleotides here. When we talk about nucleotides, we are actually referring to two major types, one of them being deoxyribonucleic acid, in short DNA, and ribonucleic acid, in short RNA. Each of these nucleotides, they are made up of certain bases. What we can, whenever I talk about the bases of the DNA, I usually make an analogy with the letters in the alphabet. Like 26 letters in English alphabet, there are 4 letters in the alphabet of DNA, A, C, G, T, by the way, and then you get these, and by putting them together, combinations, permutations, you can get different means of wording and then sentences, paragraphs, as the characteristics that are being expressed as part of the gene expression, which I will touch upon in my next slides. So the language of DNA is actually coded within these 4 bases, 4 letters. So each nucleotide, in addition to these bases, are made up also of phosphate molecules as well as the sugar molecules. So we tend to, in molecular biology, as I also tell to my students, they are learning at the same time, they are actually learning 2 different languages in one setting. Number one is the English language itself, and obviously everybody's native language is not English language. And number 2 is the jargon or the nomenclature that we use in molecular biology and genetics, which is another language, by the way. So yeah, it's as if like I'm speaking French and English at the same time. So we can go back and then start going through the structures of the bases of the nucleotides. Bases, we also can refer to them as being nitrogen-containing or nitrogenous bases. They are classified under 2 major categories. Number one is the purines, and the other one is the pyrimidines. The purines are said so because they have double-ring organic bases in the nucleic acids, and pyrimidines are said so because they have single-ring organic bases. And the examples to the purines and pyrimidines is as such. Under the purine classification, we see adenine and guanine as being purines because they have double nitrogenous bases forming these nucleotides. And in the case of pyrimidine bases, we see thymine, uracil, and cytosine. Again, these are the letters in the alphabet of DNA or nucleotide or bases, as I was just saying. In addition to these, obviously, we have the phosphate groups. They are capable of making chemical bonds for oxygen molecules with the 4 oxygen molecules. Additionally, we have sugars, pentose sugars. Penta in Greek means 5, so it refers to the ribose or deoxyribose sugars, and I will explain you in a minute, for those of you who are not familiar with these wording, what they mean. So here you're looking at the basic chemistry of the nucleotides. On the left-hand side, you see the structures of the bases themselves. The adenine here is, which is a purine, part of the purine, is double ring here. When I say double ring, I'm referring to the benzene rings in the structure. And then guanine, the same thing. Also, the pyrimidines, they are single rings, and they are as you see them here. Additionally, as I was saying, you look at the pentose sugars, you see the deoxyribonucleic acid component of the deoxyribose sugars. Deoxy because at the 2', and I will tell you in a minute what I mean by the 2', at the 2' carbon, you see that there is an H instead of an OH, where this is replaced with an OH on the ribose sugar. So that's one of the major differences between the DNA and RNA. And also on the right-hand side, as I was saying, phosphor element is capable of making 4 bonds, and you see it as here. What you're looking at here are, instead of triphosphates, is the monophosphate. A single phosphate molecule is being shown here, together with the basic chemistry of the nucleotides. Adenine, guanine, cytosine, and thymine is being represented here. I'm not sure if you are able to see this, but there are numbering going on the pentose sugar, and I'm pointing at them with my cursor. So here is carbon number 1, carbon number 2, but we don't say 1, but we say 1'. Why do we use prime numbering? Why do we have a different wording for this? This is just merely for the reason to distinguish the carbon atoms that are located in the base structures of the ring structures of the carbons, as opposed to the pentose sugar component of the carbon. So we don't say prime numbers for the carbons present in the base structure, but we use those priming so that we are also able to distinguish. So going like this, 1' carbon, 2', 3', 4', and 5'. So this priming is really important, as I constantly remind my students, because we always talk about the direction of gene expression, the direction of translation. So it's always from 5' towards the 3'. So if I haven't talked about these, you might have wondered that where does this prime number come from? Why do we say 1', 2', and whatnot? So this is where it comes from. So we need to make sure that, you know, once we're trying to build the foundation of a house, we need to make sure that you understand what we are referring to. Going along the line, we already touched base upon that the nucleotides are joined by covalent bonds in order to form polynucleotides. We have the polynucleotides, which are polar molecules with phosphate groups at the 5' and sugar molecules at the 3' end. So I will share with you in a minute some structures where it's going to be more understood what I'm talking about, and here it is. So this is the structures of both DNA and RNA. So the major distinguishing factor between the DNA and RNA, you will notice on the slides, is that the 2' OH presence in RNA and 2' H presence in the DNA molecule. Another distinguishing factor between RNA and DNA is that DNA has thymine, and RNA has uracil replacing this. So these are the major factors. Unlike the information in old school textbooks, I as a student was taught that, okay, RNA is a single-stranded entity, but we for sure know that nowadays, coming from the COVID era, we know that the coronavirus was double-stranded RNA molecules. So RNAs also can exist in double strands in nature. Let's change the gears and move along as to the finding about the double helical structure of DNA itself. In the picture, you see Watson and you see Crick here. I'm just kidding. This is myself. We had an occasion in the South Cyprus where Watson came to deliver a talk and we got invited. So that was a historical moment for us to actually be able to take a picture with this Nobel laureate. So they constructed, Watson and Crick constructed the model of the DNA structure, and they actually incorporated the information from other people's experimentation, such as the part from the chemical studies was Chargaff's experiments. The part from the X-ray crystallographic work came from mainly from Franklin's experiments. So here is the picture of like coffee cup reading style. Obviously, the experts of this know where the carbons lie and how is it that, you know, they determine this double helical anti-parallel structure of DNA through the X-ray diffraction photography. You know that, we all know that, unfortunately, Rosalind Franklin lost her life because of having, I think it was breast cancer, because they didn't know the strength of these radioactive elements when they were working with them. They actually carried the tubes in their lab coat pockets, and having been exposed to so much of X-rays, she actually passed away at an early age, and she couldn't receive a Nobel Prize because of that. The DNA molecule, as I was saying, some of these things are purposefully repetitious because I would like you to get the idea. If I'm going a little bit fast, then you can actually understand in the coming slides as me repeating myself again on the same topic. So Watson and Crick, when they proposed that DNA had two polynucleotide chains, and these chains were oriented in opposite directions, being held by the hydrogen bonding and complementary bases in opposite strands, they were describing the structure of the double helical structure, having the sugar phosphate backbones outside and then the bases facing towards inside, which actually gives us another look from a window, meaning that hydrophobic part of the double helical structure faces towards inside, which are the nitrogenous bases on which are the information is kept, and the polar parts on the backbone of the DNA, sugar phosphates. So this model actually, again, it's composed of two polynucleotide chain, as I was saying, running in the opposite direction that are coiled to form the double helix itself. On the backbone, you have the sugar and phosphate, as I just mentioned, and facing towards inside, you actually have the bases. So having talked about the base pairing, through the Chargaff's rules, we know that there is a complementation between these bases. Adenine is complementary to thymine, and cytosine is complementary to guanine. And these dots actually represents the numbers of covalent bonds that are made, hydrogen covalent bonds that are made in between these two complementary bases. Here it is, what I was actually explaining you in mouthful wording. Watson and Crick, they built this DNA model on X-ray studies from Rosalind Franklin's experimentation. And as you may notice, there is an arrowhead pointing down and arrowhead pointing up, which actually shows you the direction of the two opposite strands of the DNA running in the opposite direction. And you can also see this running opposite direction from the way that the oxygen molecules on the pentose or the deoxyribose sugar is facing. The one on the left-hand side is facing up, the other one on the right-hand side is facing down. This is another indication that two parallel strands of DNA actually are running in opposite directions towards each other. Another model of DNA from Watson and Crick, you see here the double helix here, and you actually notice that the sugar phosphate backbone is being labeled here as S and P, and then the when you see this enlargement of this picture here, you can actually see the parallel double helical running. And then specifically, I would like to touch base on the phosphate groups, which actually they form between two nucleotides. They form something called phosphodiester bond. So these phosphodiester bonds are the bonds, when broken in the triphosphate medium, they give out energy. In our old textbooks, we tend to be taught that only the ATP is kind of giving out energy, but that's not totally true, because all the other bases, we can name them as CTP, GTP, TTP, UTP, they also are able to give out energy, because they also have phosphodiester bonds, which when broken from outside towards the inside until a monophosphate is left, you can actually obtain energy for whatever the situation is to be used in the cellular systems. Here's another representation showing you the structure coming from chromosome. You can see here the chromosome itself. I'm kind of changing gears again just to have a connection with my second talk. So what this actually shows you is that you may wonder, or you may have wondered before, that how on earth it is that in humans 46 chromosomes can actually fit in this small content of the DNA, such a small volume that you cannot see with naked eye, and then it comes out to be folded, and then due to its long size, it can still fit into the nucleus. And we're going to be talking about how it does that. Here, you see a karyotype of the human chromosomal content. I usually ask my students, what is the gender of this individual? Obviously, there's a big Y here, and you should know that it's a male entity, a male individual. It represents a male gender here. So you see that the autosomes going from 1 to 22, each and every one of them, although you cannot read the small pointed writing here, they all have individual number of genes that they each represent. So what I'm trying to tell you is that the genes, or for example, that are aligned on chromosome number 1, under regular, normal, healthy individual conditions, you cannot find these genes on number 6, number 13, 18, 22, X or Y, because they are only being represented in the chromosome number 1. I will explain this to you in a minute again as to why that is. So let's kind of go over once more the properties, the important properties of the model that DNA actually illustrates us. Number 1 is the information carried in the bases. So this is very important. We should know that the genetic information in the DNA is carried in the nitrogenous bases. There are complementary strands which provide mechanism for the DNA replication, although within today's talk, I'm not going to go into detail of the DNA replication, but the structure of DNA, the way it is represented, is really important for this matter. And each strand serves as a template for a new double helical strand. Again, this is a topic of replication of DNA. This might be a talk of another symposium or another seminar. So I have said enough on DNA. Let's very quickly go over the RNA, although I did touch base upon RNA as well. But the distinguishing elements on the RNA structure, as I was saying, is that the 2' OH region and the presence of uracil instead of thiamine. Here again, the content of DNA and RNA, and there are differences you can actually observe in another different kind of illustration from this table. Having gone through this information so far, and then having actually sequenced the human genome in 2001 and 2003 in its entirety, there are things that some scientists who actually had their own biotechnology companies or some small companies such as this MyRight Genetics They tried to patent two important genes which actually cause breast cancer, BRCA1 and BRCA2, but it was then turned down that the genes that belong to everybody, every human being that has these common things, which are product of nature, are not patentable. So it was declined. But there are two questions that I can ask you in terms of whether, you know, whatever the answer, I cannot hear my colleagues' answers, but there are students in this room, they can respond to my question. Does isolation and purifying a gene allow it to be patentable? Does it? If there is a specific technique, techniques can be patentable. So in order to be able to use that technique or purchase the kit for you trying to work on a specific gene, you have to pay for it. So that's the issue about the patent. You pay for the ownership, okay? How about if the genes can be patented, what does this mean for the genetic testing? If there's going to be a genetic testing, obviously you have to pay for the testing, not for the gene itself, because the gene is present in everybody, right? So genes themselves, in other words, products of nature are not patentable. So, as I was saying in the karyotype picture, each human chromosomes, they contain a single DNA molecule. Whatever DNA molecule is present in, let's say, chromosome number 5, cannot be found in chromosome number 7 and other numbers of somatic chromosomes. They are unique, in other words, unless otherwise there is an aneuploidy or some abnormality in the chromosome, which is not the topic of today's lecture. Other than that, in a normal, the word normal is relative, obviously, but unless otherwise stated, each human chromosome contains a single DNA molecule. Each DNA molecule is extensively coiled, and it allows it to fit into nucleus, as I was saying, and this is again a representation just to clarify in your mind that, well, this is like oversimplified picture, obviously, but this is how it may look. You have the double-stranded DNA, unique double-stranded DNA in a single DNA, in a single chromosomal illustration here. So I'm going to shift my gears and then start the next part of the, I'm trying to share the slides with you. Just bear with me, please. We're going to be talking about the packing of chromosomal DNA. So if you bear with me, it will open. So I try to put you on the right grounds in terms of the foundation of the information that is governed around the DNA and RNA. Now we're slowly trying to build as to how on earth is this structure able to fit into a content which you cannot visualize by naked eye. This is a representation of a chromosome, a picture taken under electron microscopy, and the reason why I actually expose this picture to you is that you're going to start appreciating as to how it is that we can actually take this big phenomenon and then put it into small units as to explain you how things can fit into nucleus. But before doing that, we need to get to know some jargon, nomenclature regarding these issues. So the structure of nuclear chromosomes involves what you know as chromatin. So there's another big mistake which is made in the lab sessions of beginners molecular biology labs, that whenever the students are looking under the light microscope and observing onion root tips, the stages of mitosis, they see these entities, let's say in the interphase, prophase, metaphase, anaphase, telophase, and whatnot, and they think that they are looking at the naked DNA, which is not the case. The DNA in its nakedness cannot be visually seen under light microscope. You need a powerful microscope maybe to see that, or chemically you can identify that. Whereas what you actually see is the chromatin. And the definition of the chromatin is in a short, like simple arithmetic way of explaining chromatin, chromatin is equal to DNA plus proteins. We're going to expand this and explain you what proteins there are. And then the main ones were in majority found in contact with the DNA are called the histone proteins. The histone proteins can be divided into two categories. Number one, the core histones, and I will tell you what they are. And there is another linker histone, and I will actually mention you what that is as well. So if you look at the left-hand side picture here, you have a compact chromosomal illustration. If you think of this as some sort of ladies will know this, you know, when you try to knit some cardigans or whatnot, you get these wools and then you start kind of using the knitting and then you start making a cardigan or something. But you, from time to time, you need to pull out the string to make it free so that you can work with the needles on it. So similar kind of fashion. The gentleman may not understand what I'm saying. Well, I'm a gentleman. I had a lot of women around me, so I actually from first hand observed them how they did that. So if you start pulling the DNA out of the chromosome, the first thing that you're going to come across is the condensed, tightly coiled coils. This is what that is, okay, here. Usually in a class when I am teaching, I expose my students to a cord or an old kind of a telephone, which is very nicely illustrating this coiling of the DNA itself. If you keep pulling it where I'm pointing at with my cursor, you get the coiled coils being unwound, and then you see them being organized in a cylindrical fibers. On the right-hand side of this, you see the real picture taken under electron microscopy, which actually tells you that we are not making this information up. It really does exist. If you keep pulling it further down, you're going to be seeing some structure that we can call beads on a string. Each bead represents a nucleosome, and the string is the naked DNA. There is another thing that actually is analogous to this, and I tell my students, most of us, I think in Farsi and in Turkish, we use the same similar wording, tesbih. You know, when we are praying, you pull the numbers of the beads so that you can count how many times you are actually repeating yourself when you're praying. So the same thing here. You have the beads on a string, each bead representing the bead itself, which is the nucleosome, and each string is the part of the naked DNA. Okay? So for you to appreciate again, I'm going to expose you to these kind of questions. The length of the DNA, as you can observe here, and although I'm just showing you a single chromosome, imagine 46 of these fitting into this compartment, and then when you imagine about 46 of the chromosomes here together, you may wonder how on earth is it going to fit into this small, what I'm pointing at, inside the nucleus. We are now going to start discussing this, although I have started the discussion, but now you're going to appreciate it further. Just to give you a common sense of what we are dealing with, within the chromosome family of the humans, chromosome number 21 is the smallest chromosome. Okay? So you have 22 autosomes. 21st is a special chromosome, actually, because we tend to, I think it's the 1st of March, 21st of March, we tend to celebrate the Down syndrome day. So in Down syndrome people, they tend to have 3 copies of this chromosome number 21, and it happens to be the smallest chromosome in our genome. The length of it in terms of nucleotides is about 48 million. This is equivalent to 1.5% of the whole total DNA in our genome, and it is also equivalent to 10 times as much as the E. coli bacterial chromosome. So these figures, these numbers are giving you the sense of how big or how small of an entity it is, and then you can appreciate the information that how is it that these 46 of different chromosomes can fit into a small compartment of nucleus. The length of this totally comes up to be 14,000 micrometers, and as I was saying at the beginning, there is a way, there are several hierarchical organizations to which this packaging is enabled, and here it is. If we start with the whole chromosome here, you can say that a diameter has been measured as being 1400 nanometers. If you notice, we're talking about minute sizes now, because in molecular biology and genetics, not only the volumes, but the measurements are also at the level of micro, nano, pico, okay? So these are very sensitive volumes. So when you are calculating molarity and you need to know what a mole is, if you're making a mistake in terms of resuspending a lyophilized, for example, primer, those of us who work in the lab can appreciate this information better, but those of you who are new students and you're being introduced to the lab, if you're given a primer in a lyophilized state and you're being asked to dilute this because you're going to use it in the PCR reaction, if you do the wrong dilution, then you throw all the reactions away. So you need to know your simple math. So going back to this, 1400 is the diameter of this chromosome here at the metaphase stage. Once you start pulling it up from one another, then almost half of it, the diameter becomes like 700 nanometers. You keep pulling it up, and then 300, 30, 11, and finally you have the diameter of the naked DNA, which comes up to be 2 nanometers. So we can concentrate on the diameters of the nucleosome and the naked DNA. Diameter of the nucleosome is 11 nanometers as opposed to 2 nanometers of the naked DNA. Here's our famous nucleosome. So when I'm describing the nucleosome in the class, I do use the cord of the old telephones, wrapping it around on an object twice, just to show the students how it may be represented as a nucleosome itself. So when you wrap the cord, or when you wrap the DNA twice around the nucleosome, which is the green labeled color here, and then you release it, when you pull it apart, it has been measured chemically that it runs somewhere about 147 to 200 nucleotides, okay? So what this is that I will show you in a minute, how is it that this can be managed in a laboratory condition and how it can be done in a cellular environment. So this is just for you to realize what actually a nucleosome look like, other than the structure in the beads on a string. So let's move on. This is one of my favorite pictures because it actually shows you the core histones. It shows you the DNA wrapped around the core histones. It shows you I haven't mentioned them openly, but another histone which is actually pressing against the DNA, which is wrapped around the core histones, and it's called H1, which is the histone number 1. Then you have these green parts, which are the transcription factors, other types of proteins, which are bound on the naked DNA. So if you look at this, you can have a look at it under, like in this situation, again, this is more compact. This is the structure of beads on a string. So actually, if we exaggerate this and then color it and then draw it in two-dimensional structure, you can observe these entities as core histones, H1, the DNA, the linker DNA, and the transcription factors all together. I was actually talking about how is it that we know, or how did the scientists figure out that there is about 145 to 200 bases actually stuck on around the nucleosomes, and how did they separate this? To start with, you can actually use enzymes, and in this case, the enzyme used is the nuclease enzyme. ACE always depicts the presence of an enzyme, and in molecular biology, it's a given thing that when we put a suffix in front of an ACE, we know what the function of that enzyme is going to be. So nuclease, these would be the enzymes that would kind of digest or break down whatever the role of the nuclease is. In this case, nucleic acids and DNA in this part. So you can see that when nucleases are used to digest the linker DNA, you're left out with the nucleosome itself, which is 11 nanometers in diameter. And then now the idea is to separate the linker DNA, I mean the DNA around the core histone, from the protein of histone. So what they did here was they used high salt concentrations. Once they did use that, they can separate the core histones with the DNA which is wrapped around the the nucleosome proteins. Now I can talk about the histones themselves. So you can actually look at this like a big cheese, circle of cheese, and you divide them into pieces. So you have histone H2A, H2B, H3, and H4, but 2 of each. So altogether, they make up an octamer, which refers to 8 of them altogether. Octa refers to the number 8. So you have 2 of each of these histone core proteins, H2A, H2B, H3, and H4. So these make up the core histone proteins. Up till now, if I hadn't mentioned about the H1, you would have wondered, or you would have asked me, where on earth is H1? Because I haven't said anything about H1, and it is not part of the core histones, but I told you it has a role in terms of squishing the DNA, which is wrapped around the core histone, so that it cannot move out. It can only move out when there is a need for transcription, okay? So I jumped the gun and I have already mentioned about these. Let's move on. We talked about chromatin as being an arithmetical outcome of DNA plus proteins, right? And these proteins are the core histone proteins. So each chromosome consists of a single molecule of DNA complexed in equal mass of proteins. Together, DNA and the other nucleotides associated to be given the name as chromatin, as I was saying. So there are some specialties about these structure of the basic protein, which are the histone proteins. They are charged positively thanks to the presence of amino acids such as arginine and lysine due to the hydrogen ions. These are like little bristles. When we say bristles, it's like a cat whiskers. They are sticking out of the core histone proteins, and they are able to be charged positively. And because DNA is negatively charged, there's a strong pulling of each other, so that there's a tight, compact structure. When we talk about nucleosomes, you have the core histones, you have the DNA being wrapped around it twice, and then because of these positive charges, polarity of the core histone proteins, it is a compact structure. It doesn't get loose, it is under control. So here's another type of illustration of the nucleosome. Again, listing here the core histone elements and talking about the length of the wrapped DNA around each core histone proteins. Now, again, there are other explanations as to we give them specific names. Here you'd see as the title, euchromatin, heterochromatin. They both actually refer to something special. So when we talk about, when we stain the chromosomes, there are regions along the chromosomes that get stained darkly as opposed to being stained lightly. The dark regions that are stained heavily are the regions that are more compact. And we refer to these regions heterochromatic regions. I will give you examples as to what are they. And the parts of the chromosomes that are actually stained lightly, they are the regions that does not absorb a lot of dye because they are not as compact as the heterochromatic parts. So there's a reason why they are like that. And then the reason is, or the reasons are, the dense regions, which are called heterochromatic regions, are the regions where there is less number of transcription going on or no transcription at all, okay? But as opposed to heterochromatic regions, euchromatic regions are the regions where there is high expression levels because they are more open. They allow the entrance or the binding of RNA polymerase II to initiate the transcription of whatever gene needs to be transcribed. So as I was saying, the examples towards the heterochromatic regions lie in this list here. So you can see centromeres. You remember, like the structures of chromosomes, you can have them either in the middle or half, 1/3 of way through or towards the ends. So depending on where they are still centromeres, but what they make them specific is that they are heterochromatic regions where there is not much of transcription going on. Telomeres, again, the tips of the top and the bottom tips of the chromosomes. Transposons, these are the jumping genes, and junk DNA. So all these nomenclatures refers to the parts of the chromosomes or the entities where there is little of transcription taking place, as opposed to the euchromatic regions. So you might say, okay, if there is not much or very slow transcription taking place, what happens to these? How is it that these parts are being synthesized? Well, I will talk in the next session about cell cycle, and in the S phase, synthesis phase of the cell cycle, these things, the heterochromatic regions, are replicated late towards the end of the S cycle. So everything in fact, in 2003 was the time that these heterochromatic regions were being sequenced. They didn't have the sequencing of those regions, so it's a little bit more than 30,000 genes that we know of regarding the Human Genome Project. So, as I said, because transcription is not fulfilled, the genes that are relying within the heterochromatic regions are inactive, obviously. But there are other reasons that why the genes can be inactivated or stay in the inactive state. One of these reasons is methylation. In other words, addition of CH3 methyl groups to the cytosines, CpG islands. So every gene has a promoter region in eukaryotes, okay? So I will talk about, again, gene regulation in the next class after I finish this, after 10-minute break. And then I will show you where the promoter region is, what is involved as the sequence of the promoter, how is it that RNA polymerase II can bind there and then initiate from 5 to 3' direction, the transcription process, and so on and so forth. So another reason why a gene can be inactive is to be methylated. In other words, in the promoter region of that gene, CH3 groups are being added. So these addition of these bulky groups prohibit the binding of RNA polymerase II, which is like a potato. It's like a big entity, enzyme, protein, which actually needs to be able to bind with the aid of other transcription factors to the promoter region. But if the promoter region is blocked by means of these CH3 groups, there is no place that RNA polymerase II can bind. So that means that you're going to have an inactive gene. Unless otherwise something happens, then they are reactivated, okay? So we touch base upon the binding of transcription factors to the promoter. They are usually the initiators of transcription, which they welcome actually. I kind of make analogy between a queen and her helpers that actually, like, when she wants to sit on her chair, they kind of show her the way, they welcome her, they help her out. So the transcription factors are also like that. So I refer to RNA polymerase II as a queen of the situation, because the enzyme itself cannot go and directly bind to the promoter region, but it requires some transcription factors to bind first and then kind of welcome the polymerase enzyme to come in and then initiate whatever the job it's going to be. So this is the last slide that I want to share with you under this lecture. So you see on the left-hand side, two-dimensionally drawn illustrations of how the DNA and chromatin rather can be compacted and fit. And on the right-hand side, just to make you believe, if you think that I'm talking nonsense, these are real pictures taken under electron microscopy, okay, which match to the illustrations that we're trying to show you to make you believe or to make you realize and appreciate how the DNA double helix can fit, can be packaged into the nucleus. Thank you, guys.